Gene expression means how the code that is encoded in DNA will turn into a protein. The central dogma states that DNA is the coding strand, and it's going to get transcribed by RNA polymerase and forms messenger RNA that carries information to the ribosomes, go through translation and forms protein. This is the central dogma. And pretty much every organism is doing it, but sometimes RNA, some virus has RNA, and they use reverse transcriptase, that they turn their RNA into DNA and messenger RNA and protein to execute the code what they have. Gene expression happens slightly differently in prokaryotes than in eukaryotes. We will just briefly state the prokaryotes, that is more simple than the eukaryotes. So one gene is coding for one polypeptide, one protein. Both the transcription and translation happens in the cytoplasm, and the transcription and translation happens simultaneously in the cytoplasm. In eukaryotes, on the other hand, one gene can code for many polypeptides because the messenger RNA goes under processing. So the transcription happens in nucleus and translation in the cytoplasm. Transcription, we will focus on the transcription in eukaryotes, starts with initiation. We have two DNA, we have the DNA, the double helix. One is the 5' with the phosphate at the end, 3', they are anti-parallel, and the 5 to 3 is the coding strand, the non-template strand, and the 3 to 5 is the template strand, the template DNA. There are segments on the DNA that the promoter where starting of the coding starts, the reading part, the transcription unit that transcribes for polypeptides and two different RNAs, transfer or ribosomal RNA, and the last one is the terminator part. The promoter can bind many transcription factors. These transcription factors are proteins, and they help the RNA polymerase to bind to the promoter side. In prokaryotes, they don't have transcription factors. Now, with the transcription factors, the RNA polymerase can bind easily to the promoter, and both the transcription factors and RNA polymerase all together, they form the transcription initiation factor. Now, with this factor, the RNA polymerase can go and do the transcription. So this is the initiation. Good. So the elongation is happening. The messenger RNA is going to form in the 5 to 3' direction. 5' is over there, 3' is close, and it's adding all the time to it. Pre-messenger RNA is the same as the coding strand, but with different nucleotides. Instead of the thymine in the RNA, we have the uracil. So the elongation is in 5 to 3 direction. And the last step of transcription is the termination. The termination is when the RNA polymerase reaches that segment, it will get off from the DNA, and the pre-messenger RNA is getting off from the DNA template. After we have the pre-messenger RNA, it will go through post-transcriptional modification. This does not happen in the prokaryotes. In eukaryotes, that's why one gene can code for many polypeptides. Here's the pre-messenger RNA 5 to 3 prime, and a cap is going to be added to the 5 prime, and a tail polyadenine tail will be added to the 3rd prime. It has some segments, introns, exons, introns, and exons. I stands for intron and E stands for exon. And the splicing, just cutting out the introns, is by a spliceosome. It's an enzyme that contains small small nuclear RNAs and proteins, and they act as catalysts. So it's not only the protein that can act as a catalyst, but the RNA molecule, so there's a ribozyme. So we are taking out the introns, they stay inside, and the exon is ready for exiting out the nucleus. We have the caps and the tails, and now the exons are spliced together. Here is the nuclear envelope, and the matured or processed messenger RNA is going into the cytoplasm for translation. Translation happens in the cytoplasm in eukaryotes. Here is the messenger RNA now, 5 to 3 prime. We have the caps and the tails, and these are the nucleotides. I tried to draw it, and they come in triplets, and they are called the codons. The coding is redundant because the 4 nucleotides can give 64 combinations, but we have only 20 amino acids to code. So more than one codon codes for the same amino acids. Here's a transfer RNA transferring the amino acids. So it has AUC over there. This is called the anticodon, and to combine with amino acid like methionine, it needs energy. ATP is providing the energy to combine the transfer RNA with the amino acid. Here's another transfer RNA that has different anticodons, and it combines with another type of amino acid. The anticodon is very specific what type of amino acid is combined with the transfer RNA. So here is a small subunit of the ribosome, and above it, it's a bigger subunit. That's the big subunit, and it has some sites. This is the P site, the E site, as well as the A site. The P site stands for the polypeptide formation. Yeah, so the ribosome is moving to the right and the messenger RNA to the left. This anticodon is going to bind with the first codon through hydrogen bonds, and probably I have to just include that for the A site, and this will contain the A site. So this is the other transfer RNA is going to just combine at the A site. They drop off their amino acids, and peptide bond will form between the 2 amino acids. I have a good video, and I will show you guys in class to visualize the process. The elongation and the termination. Termination will happen when a stop codon will be reached. it does not have any amino acids. It's just really nothing. And the folding and modifying of the polypeptide will take place. I just draw a sequence of a polypeptide, a short one. It starts always with methionine, that's the starting codon, but the methionine sometimes is not present as a first because it acts as a signal peptide and tells the peptide where to go in the cell, for example, going into the endoplasmic reticulum for modification. Mutation and the gene level, very fast. The substitution of the nucleotide in the DNA can cause silent mutation because of the redundancy of the coding, as well as it can have missense mutation. That's another amino acid will be coded instead of the original, and nonsense mutation, early stop codon. Insertion and deletion will lead to frame shift mutation, and it's really bad. Mutagenes can cause gene level mutation. It can be physical factors or chemical factors. Physical factors are X-ray or UVs, and chemical factors are, for example, smoke that affects the DNA structure. That's the end.